Okay, everybody, it's 6 o'clock. So, in an effort to have a full hour of exciting information and question and answers, I want to get started. My name is Dawn Eaton. I'm the CEO of Susan G. Komen Central Tennessee, and we are just delighted to have everybody on the call tonight. This is our first ever forum, if you will, or panel about breast cancer research. And so it's not going to be our last. And we're really, really excited to have everyone on tonight. I really want to give a special shout out to our partners and sponsors tonight. We have LifePoint Health, Nashville Yards, Immunomedics, Pfizer Oncology, and Seattle Genetics. So I know some of them are on the call tonight, and I just want to give a special shout out because this wouldn't have been possible without them. We're just going to start with a couple of housekeeping just topics that we want to cover. If you will, be sure to keep your mic muted if you're not talking. I wanted to make sure everyone knew that our program is going to be recorded. So once this is over, we want you to feel free to share this. We'll be sharing it on social media and via email, and we want you to feel very comfortable and share it with other folks, your friends, your colleagues, etc. If you need any technical assistance, Madeline Burgess with our office is on the call. Her contact information is in the chat box, both her email and her phone number. And just a little bit about the format for tonight's presentation. We're going to be doing a Q&A led by Dr. Jennifer Pietenpol, our moderator. And so we'll be covering 3 topics: deciphering and treating breast cancer, new and on the horizon breast cancer therapies, and updates on breast cancer therapies. So you're in for a treat. And then we'll have Q&A at the end. So we're reserving the last 10 minutes for questions. And I'm going to ask if you will put your questions in the chat box. And then Madeline will be reading those and going over those at the end of the night. And we'll be sure, and if anything we do not get answered, we will make sure that we answer and get back out to everybody. And I just want to invite you to keep your video on if you'd like to. We'd love to see your faces, and especially when we get to the question and answer part. So without further ado, let me just tell you a little bit about our mission. We always like for everybody to know what we're about. So Susan G. Komen Central Tennessee is one of 61 affiliates across the country. And our mission, and this is our mission nationally, not just for Central Tennessee, but it's meeting the most critical needs in our communities and investing in breakthrough research to prevent and cure breast cancer. And that's where our topic and our emphasis will be tonight, is about the research. And with that, I'd like to introduce our speakers tonight. You all are in for such a treat. We just have the most distinguished panel of scientists in the nation. And I'm really happy to tell you they are all in Nashville. So we're very, very lucky. First of all, we have Dr. Skip Burris, who is president and clinical operations and the chief medical officer at Sarah Cannon. We have Dr. Ingrid Mayer, who is the co-leader of the breast cancer research program at Vanderbilt-Ingram Cancer Center and also a Komen scholar. We have Dr. Ben Park, who is also the co-leader of the breast cancer research program at Vanderbilt-Ingram Cancer Center and another Komen scholar. Dr. Erika Hamilton, who is the director of the breast and gynecologic cancer research program at Sarah Cannon, and also one of our Komen Central Tennessee board members. And we also have as our moderator tonight, Dr. Jennifer Pietenpol, who is the director of the Vanderbilt-Ingram Cancer Center and the co-chief scientific advisor for Komen on a national level. So with that, let me turn it over to Jennifer, and she'll start our panel discussion tonight. Thanks, Jennifer, and thanks to everybody for being here.
Thank you, Dawn, for that introduction and introducing this really amazing expert panel that we are fortunate to have with us this evening. And I just want to thank everyone here from this area and this region for joining us tonight. We know everyone probably has a lot going on, and also at times there's Zoom fatigue. So for all of you to really tune in for this means a tremendous amount. And we're going to highlight recent advances in breast cancer research and care. And you're going to hear about new approaches being used to decrease breast cancer mortality, even more than the tremendous progress we realized over the past 10 years. As Dawn mentioned, the panel comprises truly the world's expert in breast cancer, and we are fortunate they live, practice, and conduct research right here in Middle Tennessee. And they're quite frankly changing the practice of breast cancer care, both within this region, but importantly around the world. You'll hear about effective and targeted therapies and more individual or personalized treatments. And we have all of you, Komen Central Tennessee, and to thank for supporting the paradigm shifting research that you're going to hear about tonight. And this has really changed care for patients and families facing breast cancer. There is so much work to do. Your support is more important than ever. You know, 250,000 people a year are diagnosed with breast cancer in the US. 150,000 are living with incurable disease. And 41,000 women and men are dying every year, US alone, of the disease. So there's still a tremendous amount to do. We're going to gain insights from the experts tonight on approaches to really take on this challenge. And why don't we go ahead and start by talking about deciphering and treating breast cancer. And so we're going to start with Dr. Erika Hamilton, and she's going to talk to us briefly and set the stage with the different types of breast cancer, which ones are the most challenging in terms of treatment and survival, and really where tremendous focus has been. Erika?
Thanks, Jennifer. So there's different ways to answer this question, actually. There are two kind of overarching types of breast cancer that we traditionally talk about, either ductal or lobular, and there's a few kind of rare caveats to that. Ductal is where the cancer starts in the ducts or the tubes that are in the breast. And then there's also lobular, where the cancer starts in the lobules, or those where milk is produced in the breast. But most of the time when people ask this question, they're really talking about the markers of the breast cancer. So, the markers really are ER, PR, and HER2, and that stands for the estrogen receptor, progesterone receptor, and HER2, which is a protein that tends to be a little bit more aggressive. So we commonly talk about three different types of breast cancer: the hormonally driven breast cancers, or HR positive, that's estrogen or progesterone receptor positive; the HER2 positive breast cancers, or this third category called triple negative, meaning it does not express any of these three receptors. So traditionally, we think about HER2 and triple negative being the most aggressive. 70% of the breast cancers are hormonally driven. Those tend to be the ones with the best prognosis, the ones that are easiest to treat. However, over the past few years, we've had a lot of advances in HER2 positive disease, new antibodies that have really improved cure rates. And so now HER2 has kind of probably a little bit more favorable than it used to be. And so triple negative tends to be the hardest to treat. Up until last year, we had no targeted agents for triple negative, meaning we really had to treat people with chemo, and more chemo. And now we have approval of a new drug, immunotherapy. So I think we would still classify triple negative as probably the hardest to treat, and triple negative and HER2 is the more aggressive cancers.
Thank you, Erika. Dr. Ingrid Meyer, what is metastatic breast cancer, and why is it there's so much focus on that form of the disease?
Thanks, Jennifer. So metastatic breast cancer is essentially breast cancer that at some point has started out in the breast, but microscopic cells that spread out and travel through the bloodstream and lodge themselves in other parts of the body. Time goes by, eventually they grew and became another niche, another focus of breast cancer, far and away from where the breast cancer has started. So this distant spread of the cancer is what we call metastatic disease. A lot of people interchangeably call that stage 4, which is not necessarily an accurate description, because stage 4 only applies for patients that present with metastatic disease from the get-go. But essentially, for practical purposes in terms of treatment, they mean the same thing. It is cancer that has spread outside the breast area and has gone somewhere else. The bigger issue with this process is that these cells that leave the breast and go elsewhere, they are a different beast than the ones that started out in the breast to begin with. They're more aggressive. They are able to survive outside the breast cancer environment, the breast environment, if you will. And because of that, they're harder to kill. And they are not, even though they're part of the same cancer, a lot of people think, aren't they all the same? They're not the same. They're very heterogeneous. So some of these cancer cells are more aggressive, some are less aggressive, some are more resistant to certain treatments, some are not. So because of this incredible difference between them, it's just impossible to use one treatment to tackle them all. So even though we have achieved a lot of successes now in controlling metastatic breast cancer a lot better than we could, eventually all we're doing, unfortunately, to this day is kicking the ball down the road, where we've improved survival, we keep people alive for longer, but ultimately metastatic breast cancer is uniformly fatal. Like, people die of breast cancer no matter what. So unfortunately, metastatic disease equals trouble, and it is incurable disease, but a lot of advances have been made in that field.
Thank you, Ingrid. And I think it's for those reasons, and because this is what drives mortality in breast cancer, that's such a significant portion of the Komen research portfolio is focused on the metastatic form of the disease.
Absolutely. It is the one thing that is killing patients. Obviously, ultimately, if we do better in prevention and treatment of early disease, ultimately that will translate in less metastatic breast cancer. But the urgent need is to help patients that are at risk of dying. So that's why, indeed, the focus of research, but not uninvariably ends up falling into metastatic breast cancer.
So Dr. Park, give us an example of cutting-edge research that is really going on in breast cancer that you think holds great promise for treating it, or perhaps even, as Ingrid just said, diagnosing it earlier so maybe it can be cured through surgery or local containment.
Oh, thanks for that. I think there's so many examples, it's really hard to pick. I do want to point out that my colleague, Dr. Meyer, if you look carefully, her dog just walked in the background, and it's one of my favorite things to do when I Zoom with her. One of the things that I and many in the field have been working on is a so-called liquid biopsy. And what that is when we take a tube of blood from patients, we can actually now count and categorize cancer DNA that's free-floating in the blood. Why is this important? Well, there's a number of different ways we can use it, but from what Dr. Pietenpol was addressing or asking, one can imagine that, as an example, you might be able to use this to diagnose breast cancers at an even earlier stage, or whether or not we could use it as an additional test to help us diagnose breast cancers when we have somewhat equivocal findings on mammogram or breast MRI, let's say. So there's a diagnostic component that I think would be very, very critical into catching breast cancers earlier, because I think most of us would agree that the earlier we catch the disease, the higher the likelihood of cure. The second area that we and others are working on now is, could we use this almost as a measure of total cancer disease burden in the body? And you can actually think about how in early stage breast cancers, we don't really know who's cured and who's not, and therefore we don't know who should get which therapies. We kind of uniform or design uniform protocols based upon clinical trials. I think in the future, what we can really start to expect with these liquid biopsies is to individualize based upon whether that cancer is still there or not, so we can follow it in real time. And then similarly, metastatic disease, having the opportunity of not only finding cancer DNA that tells us, hey, this is the Achilles heel, and we might have a drug for that particular cancer because of that DNA alteration. And again, quantitatively seeing whether or not our therapies are working in real time. That gives us an opportunity perhaps to really impact patients' lives by giving them therapies that really work and not giving them ineffective therapies that are just exposing them to needless toxicities.
Thank you, Ben. Dr. Burris, so if we can't detect the cancer early enough for surgical resection and cure, many patients go on to getting medications, either IV, or some actually now get medications in pill form. So why can't all patients now get pill forms or medication? And also, some of these are real pain, whether IV or injection. So what's the future hold? You might be muted, Skip.
Oh, there you go. Thanks. It didn't come off. So Dr. Pietenpol, Jennifer, thank you so much, and please call me Skip. It's a great question. I haven't had that conversation since a few hours ago when I was having to explain to a patient why they needed to have a port put in. This has been going on, and my career started back in San Antonio 30 years ago when we were beginning to do the phase 1 trials with capecitabine that grew up to become Xeloda, and the idea that you give 5-FU orally. And it was shown to me by the pharmaceutical company at that time that to give, until they came up with the technology to have the prodrug capecitabine, it would have been a giant sweet tart, if somebody remembers what those used to look like, which were great candies. That's what you would have been swallowing or breaking up. It's either because the molecules are too big to get into a formulation to go in a capsule or a pill, or they're too big to cross the lining of your intestines, either the stomach or the small intestine, or they're in fact an irritant. A chemotherapy can be irritating, some of these other medicines, to the veins and into the vascular system. Fulvestrant, which Dr. Pietenpol alluded to, is literally a pain in the butt. And since it came along, it's become a double pain in the butt, but an effective medicine, and so one that doctors prescribe and you patients graciously take and put up with. On the good news front, we're seeing really quick advances in terms of moving to the oral form. There's a number of drugs that Vanderbilt and Sarah Cannon both have in clinical trials, and I think the first oral type of fulvestrant will be approved before the end of 2021. So over the next 12 to 18 months, we're going to see a drug be approved in that setting. I think secondly, we're seeing some of the chemotherapeutics, much like the capecitabine 5-FU story. There's been recent positive trial results with oral taxanes. So we're all familiar with paclitaxel and docetaxel. The pharmaceutical biotech companies have now come up with a way to, in fact, get those drugs in oral formulations. And there's 4 compounds in clinical trials. So that's coming very quickly. And then most surprisingly, maybe, is the monoclonal antibodies. It hasn't been talked about a lot because it's for a very niche setting, but with the addition of an enzyme that helps aid in absorption and stability, giving the trastuzumab and the pertuzumab, the Herceptin Perjeta drugs, preoperatively in breast cancer now can be done subcutaneously. So not orally, but done as an injection under the skin. And I think we'll continue to see that technology being explored. So these are places where it's investment in formulation, investment in trying to make things easier. But I think for the patients, it's certainly the right thing to do. And I think the future is bright for having most of our drugs either being given orally or as a small subcutaneous injection.
It's truly exciting, Skip, to think how far it's come and how quickly. And it's kind of that what you can do with chemical discovery. Really right. Exciting. So let's turn now to on the horizon are new breast cancer therapies. And Ingrid, you know, we just heard Skip talk about chemotherapy and actually some anti-estrogen therapies and actually smart ways to put new therapies on biological transporters like antibodies. Tell us now about a therapy that actually is advertised in every airport for all different types of cancer. What is immunotherapy? And are there different types?
So immunotherapy, it's obviously the new buzz, the new sexy thing as far as treatment, not just for breast cancer, but for many other cancer. And it's actually an interesting concept and one that is probably misunderstood by a lot of people, where when people hear immunotherapy, they think, oh my God, this is something that's going to make my immune system strong and it's going to fight the cancer. It's not actually quite like that. So what immunotherapy does actually is make your cancer visible to the immune system. So not that it makes the immune system stronger, but it actually unmasks the cancer as something that doesn't belong there, allowing then somebody's immune system to actually attack that foreign entity in one's body. So what happens is that cancers, unfortunately, they're smart and they've figured out ways to elude, you know, anything that can kill them. So one way that they figured out is to hide from your immune system. And the ones that do that very effectively typically have a protein called PD-L1 expressed in the surface of their cancer cells. And what this PD-L1 does, as I said, think of it as an invisible shield, and the cancer just like goes unnoticed by the immune system. So there are immunotherapy drugs that will shut down PD-L1 or other proteins similar to that. So it doesn't have to be PD-L1, it could be PD-1, it could be CTL4, it could be a number of other things. So each one of these different immunotherapies will tackle different components of this protein that allow or don't allow your immune system to recognize something as foreign. And therefore, once that happens, some of these foreign proteins that, your cancer is still part of you, but it's still different than you. So these foreign proteins called antigens now become visible to the immune system. So sometimes combining, and I know that sounds paradoxical, but combining chemotherapy with immunotherapy in breast cancer actually has made a big difference, especially for cancers like triple-negative disease. Because even though chemotherapy suppresses your immune system, it actually shakes up the cancer and makes some of these antigens, some of these foreign proteins be more visible to the immune system. And when you add immunotherapy in there, now you not only have the chemotherapy attacking the cancer, but you also have your immune system doing that. So that all sounds fabulous and great, but there's two caveats to this. One is that not everybody benefits from this. Typically, at least to this day, and this is not perfect, there's still a lot of work that needs to be done, but at least to our knowledge today, only breast cancers that express this protein called PD-L1, as I said, will respond to immunotherapy. And even then, not all of them do. But also, the issue with immunotherapy is that if it works great, but it also has side effects. And the difference between side effects from immunotherapy and side effects from, let's say, chemotherapy is that even though they are uncommon, they are permanent. Once you turn on the immune system against the cancer or you, that's the gift that keeps on giving. So there are these two caveats. It's not for everybody. When it works great, but the side effects can be permanent. So there's a price with these immunotherapies.
Thank you, Ingrid. So Ben, Ingrid's leading us then down a road to realize not many patients, breast cancer patients, receive immunotherapy. It's still a small fraction. What can we do to change that? And maybe briefly, share with everyone here that immunotherapy extends to other things, adoptive, cellular therapy and vaccines, and maybe just a word on those.
Yeah, so I think Ingrid really touched on a lot of the points that make us a little bit gun shy just to use this on everyone, right? It can be very expensive, it can be very toxic, and some of those side effects quite literally can lead to death. And so I think we have to be very careful about who we choose to receive immunotherapy. And as mentioned right now, the thing that we have an approval for is in first-line metastatic disease for triple-negative breast cancer patients. Dr. Hamilton had mentioned that earlier. There is a potential another indication in use, and I think this gets to some of what you're saying right now, but it's not just for breast cancer per se, and that is something that we know now called high tumor mutation burden, meaning that in some cancers, breast, colon, others, for whatever reasons, they can have a lot of DNA mistakes in their cancers. Ultimately, cancer is a disease of DNA gone bad, and by that I mean it's full of mutations and other DNA alterations. It turns out that work done by colleagues at Hopkins really showed that the more of these mistakes a cancer has, the more likelihood they're going to respond to these immunotherapies. And so as an example, I had a patient with a specific type of lobular breast carcinoma or cancer called lobular carcinoma that we heard about earlier from Dr. Hamilton. And sometimes lobular breast carcinomas have really high tumor mutation burdens. They have a lot of DNA mistakes. And in fact, this patient of mine actually responded for well over a year, about a year and a half, and she had no evidence of disease getting immunotherapy. That has now led to the FDA approval just this year that any solid tumor, breast or otherwise, that has a high tumor mutation burden can get this because of the chance of benefit. And then kind of more on the horizon are things like cellular therapies where we can re-engineer patients' T cells or their immune cells to try to direct them to the cancer, or using even other kind of bridges. Sometimes we call them bispecific T cell engagers or other types of antibody therapies that will bring together immune cells with cancer cells. These are also on the horizon. They clearly work for other types of cancers, and that's where we're actually trying to adopt them for our particular set of diseases, triple-negative breast cancers, ER positives, et cetera.
Thank you, Ben. So Skip, you briefly touched on antibody-based, chemotherapy. What's the scope of antibody conjugates out there, and more importantly, what's on the horizon going forward for this modality of treatment?
Great question, Jennifer. And I think as Ben and Ingrid and Erika have alluded to great hormonal therapies, promise of immunotherapy for some populations. We know the HER2 success stories, but the fact remains chemotherapy is going to be an important part of what we utilize to help fight breast cancer. And so the idea of an antibody drug conjugate, so an antibody so that we have delivery to a specific cell with a linker so that the potent cytotoxic, which is the third part, the chemotherapeutic, stays attached to the antibody. And the hope here is that it gets only to the cancer cell and we have minimal damage to other tissues. And for that reason, when these first were developed in the 90s, they were called smart bombs. And those of the 90s you've never heard of, because those of us who did the trials had to close them because we weren't specific enough. It really needs to be something that only targets receptors found on cancer cells, and the linker wasn't good enough to hold the potent cytotoxic. And the potency of the chemotherapy drug matters because you can only put a few molecules attached to these antibodies. That being said, over the last 10 years, great advances. We had the TDM1 molecule that was specific for HER2 breast cancer. We have a new drug, the trastuzumab deruxtecan, that's out there now, known as Enhertu, that has been approved for HER2 positive breast cancer. And now we have the sacituzumab govitecan, known as Trodelvy, approved for triple negative breast cancer. Again, an antibody that hits a specific target on the breast cancer cell linked to a potent cytotoxic. We've gotten better tolerated chemotherapeutics that are being attached, which is exciting. And Jennifer, you asked about future targets. The ones that are coming out, really, I think the imagination of the scientists to know which ones are best. We hear about buzzwords like NaPi2b and AXL and different targets that are there. So I think this is hopefully going to be the future of chemotherapy. And the idea that by giving it in this fashion, we'll have less side effects, we'll have less hair loss, we'll have less GI toxicity, and we'll really get past some of the basic chemotherapy that we think of today.
Thank you, Skip. So Erika, let's consider one of really an amazing therapy that really changed the face of HER2 and trastuzumab and those therapies. Will those ever be replaced with different treatment regimens? So
I think that's a great question. I'll predict the future. So, you know, we have quite a few different types of therapy for HER2 breast cancer. We have the antibodies, like you were mentioning, trastuzumab, pertuzumab, which is the newer one on the stage. We also have antibody drug conjugates, 2 of which are approved, like Skip was talking about, both TDM1 or Kadcyla, as well as trastuzumab deruxtecan and HER2. And Enhertu was just recently approved in the past year, so that's very exciting. And then we also have this third class of molecules, tyrosine kinase inhibitors, and those are the pills. Those are the ones that ends in ib, like lapatinib, et cetera, neratinib. You know, I think that there potentially are new drugs outside of these classes coming. Bispecifics are one that kind of comes to mind, and that's where you can even attach 2 antibodies on the same molecule, so it kind of has a left side and a right side. And that could be something like trastuzumab and pertuzumab together. Or it could even be trastuzumab with immunotherapy on the other side. So I think there's a lot of potential new drugs coming. I don't know whether our current paradigm is going to be completely thrown out the window. You know, we really do cure very many patients that we get our hands on early that don't have metastatic disease when they present. So, you know, even if people have node positive breast cancer when they present, with the HER2 therapies now, the vast majority of these patients will be cured. I think what we'll probably get better at is finding the right size therapy for the right patient. And what I mean by that is kind of not a cookie cutter approach where we give everybody the same thing, but we get smarter at figuring out the individual characteristics of someone's tumor and deciding whether they're going to do really well with less therapy or whether they have a bad player and they may need even more therapy than what's standard.
Thank you. So Ben, let's move a little bit from these environments outside the cell, the immune system, immunotherapy, receptors, whether estrogen receptor or HER2. Now let's drill down to the nucleus and let's get brought up to speed on what it means to test someone's germline DNA and or take the DNA of their tumor and take a look at it. And what are the different tests? What are they for? And then how are they helping advance kind of either how we better treat or even prevent breast cancer?
Yeah, that's a great question. You know, I think the confusing part with cancer, unlike a lot of other genetic diseases, is that we look not only at the patient and what hereditary kind of gene mutations or alteration that they may carry and be able to pass along to their children, but we also look at the DNA of the cancer itself. And that's what I was talking about earlier when saying that really cancer is a disease of DNA gone bad. Many times we think it's largely or exclusively just in the DNA that's in the cancer, but we now know that there are families, the so-called BRCA1 and 2 families as an example, that if they carry this a mutation in either of those genes, it predisposes them greatly to developing breast and or ovarian cancers. And so the two are interlocked. They're parts of the same puzzle. But having a BRCA mutation accelerates the path to cancer, and that gives us the opportunity for prevention and earlier diagnoses, because we know that if one has inherited a BRCA1 or BRCA2 mutation, or now other genes that we can look for, we have a whole panel of genes that we now test for hereditary breast cancer syndromes, that would increase the lifetime risk of developing breast cancer in that particular carrier. Again, not even having a breast cancer diagnosis. So that brings about opportunities for earlier screening, for prevention, for prophylactic surgeries as an example, or even using endocrine therapies or hormone therapies as we sometimes call them, really opens up a great amount of possibilities simply because of knowledge. And knowledge really is power in this regard. On the other hand, when we look at a cancer, because it's from that patient, let's say the patient had a BRCA1 mutation, we're still going to see that BRCA1 patient in the cancer itself because, again, it's from the patient. But we'll also see additional genetic mistakes that have been acquired along that road from being a normal cell all the way to a cancer. And sometimes, and we're getting even more and more drugs nowadays, sometimes we have drugs that can target those molecular alterations. I think as Dr. Hamilton just mentioned, HER2 being the prototypical example. HER2 really is a genetic change in the cancer. And now we have great drugs for it, but now we have many more.
Thank you, Ben. So Erika, Ben just mentioned there's now there's panels of genes that are, you know, women and men are tested for that may be germline alterations that give an increased risk for breast cancer. So it raises the question, should germline testing be done for all women and men who get a diagnosis of breast cancer at the time of diagnosis?
A good question. You know, so right now, you know, breast cancer unfortunately affects 1 in 8 women. So the majority of breast cancers are not linked to the BRCA1 or 2 gene. And then we have this kind of gene that we've learned about a little bit more recently, PALB2, which can be linked to breast cancers as well. So right now, you know, families that have a very strong history of breast or ovarian cancers, genetic counselors actually kind of fill out a family tree and figure out, you know, how many people are in the family and how many have had cancers. There's an algorithm that goes into kind of whether we would recommend genetic testing there. What's new over the past couple of years is that we've had this new class of medications approved called PARP inhibitors. And these are pills. And they're approved for patients that have BRCA1 or 2 alterations. And patients that receive this pill actually did better than chemotherapy, which was the standard. And so now for patients with metastatic disease, meaning disease that spread beyond the breast and the armpit out to other places in the body, that's indicated for anyone. So any patient with breast cancer that is HER2 negative qualifies for these germline BRCA1/2 tests, and a decent percentage of patients with early breast cancer based on the family history. But, you know, the average patient that comes into clinic who may be 60 or 70 and doesn't have a whole lot of breast cancer in the family, that's probably just good old fashioned bad luck cancer, the 1 in 8, and not necessarily familial linked.
Thank you so much. So Skip, back to you. There's so much in our lives that we are actually probably subjects of experiments that are happening with data engineering and big data learning. People know when we shop on the web. People know our preference in buying a car already from other data they've collected. So it raises the question, when we look across everything that is now accumulating in terms of health data, will machine learning or artificial intelligence, what will it do for breast cancer? You know, will it be able to prevent or detect breast cancer earlier in populations and individuals in the way it's being used to predict other behaviors?
You know, the possibilities here are just mind-boggling. I know it wasn't too many years ago we worried about machines replacing humans, and now we're at a place really where, with everything going on, the machines are really giving us 4 or 5 hands, maybe a dozen hands to work with. And credit to the Vanderbilt program has a world-renowned precision medicine data analytics platform. The people of Middle Tennessee probably aren't aware of what a gold mine that is with regard to the trust placed by the government in Vanderbilt to really process a lot of that data. We have made an investment at Sarah Cannon in a company called Genuspace, which is a analytics platform, computational biology. Sounds fancy, not sure what all that means, but all of it is teaching machines to be able to think like humans think faster and to consume much more data. Thinking about, you know, all the books you'd like to read, and, you know, maybe you get through 1 a week, and if you do a machine correctly, they can get through thousands in a week. And so that's the power of what we're talking about. And as Jennifer alluded to, we need to think about where this can help out in breast cancer. It's all over the place, where there's a company in Boston that is looking at rereading mammograms with being able to look at the density and the particles that are part of that image, and looking at it in a way that the human eye can't do fast enough to really look at a better way to see what's suspicious on a mammogram, maybe before the human eye can pick it up. There was a fascinating study in pancreatic cancer done in Western Europe, mostly out of the UK, where they looked at newly diagnosed pancreatic cancer patients, and when they assimilated all the data from those patients' records over the past 5 years, you could tell what group they predicted with very high sensitivity who was heading toward pancreatic cancer from very subtle things like gradual weight loss and changes in their labs with regard to protein. So where this will go in breast cancer, I think one's going to be drug discovery. I think, as Dr. Park was alluding to, Ben mentioned all the DNA sequencing we're doing, beginning to understand what are new targets to treat breast cancer with. So that's one of the places where our chemists and scientists are working together. I think secondly, we're trying to accumulate this data to look at, with the combination of some of the circulating DNA in the blood and mammograms, what might be predictive that could come up in somebody's blood work. There's a trial that'll be a while to report, but a company called Grail that's looking at early detection by liquid biopsies, by blood samples, took over 100,000 women getting mammograms and took a tube of blood from them and sequenced them. And they're going to give a tube of blood each year. And we're going to be able to look back, again, aggregating 100,000 patients with what will be 500,000 mammograms, which will be billions of DNA sequences when it's all added together, to see if we could predict that when you go in and get your mammogram and you get a blood draw, and this might be the future just a few years from now, your level of likelihood to be looked at more closely or what problem might be developing will be part of the story. So it's a long answer. You could talk about this, but the hope of big data, of machine learning and artificial intelligence, I think is really going to improve survival in cancer across a number of diseases.
So Ingrid, if we now, you know, we've had a nice discussion on new technologies, new ways to detect cancer, amazing discoveries with immunotherapy, and Skip kind of talking about how we could potentially bring this all together for even better ways to predict the disease or treatment. The reality is, and this was magnified during the pandemic, there are tremendous health disparities. So we now know that many of the treatments that are discoveries that Komen has funded the research for don't make it to the right patient population. And so what are some approaches we should consider for addressing the significant health disparities that exist in breast cancer? Many of us realize that of those 41,000 deaths a year, likely 25–30% of those are probably unnecessary deaths, meaning people did not get the right care at the right time. They did not get the correct diagnosis of the disease. What are some approaches we're taking, but also what's the future hold? Because this is such a critical area.
Thanks, Jennifer. So because we don't have 72 hours to discuss this, because it's an incredibly loaded and huge topic, I'm going to try to summarize a little bit in buckets. So when we think about health disparities, there from a research point of view, there's so many hits that come in our minds all at the same time. One of them being socioeconomical disparities. One may be racial disparities. One may be, again, access to care or age, or a number of different things that color this health disparity. So obviously, one thing that I think Komen has impacted well, and there are several examples across the country, and potentially being, I'm not going to call it a low-hanging fruit, but I guess I'll start with that, being a low-hanging fruit, is helping patients get access to care. So just making sure that people make it to their mammogram, making sure that people make it to the doctor when they have a breast cancer symptom or something that changed in the breast, whatnot. That's obviously the big duh, because the sooner we diagnose a breast cancer, the more curable it is, the less treatment we need to do to achieve a cure, and so forth. So obviously focusing efforts on prevention, early diagnosis, access to care, making sure that people have proper insurance, making sure that people have insurance that, let's say, covers their genetic testing if it's appropriate. I mean, like making sure that people are getting what they need to get their optimal treatment for breast cancer is paramount. And it's not necessarily something that I think research answers, although not research in the sense that we think, but obviously epidemiologists and a lot like policymakers, whatnot, obviously are looking at this very closely and coming up with guidelines and recommendations and making sure that people are not being dinged for pre-existing condition if they get, let's say, a genetic testing because their mom had breast cancer, or things like that are all things that are awfully important. But there's this whole other bucket, aside from this access to care, socioeconomical, blah, blah, blah issue that has to do with actually genetics. I mean, we're not all equal, and there is a lot of difference between me and you, me and somebody else, white women, black women, Asian women, and so it goes. I mean, we're really not created equal because clearly, like, medicine has shown that there is different tolerance to treatment. To give examples, right? So for instance, if you take all women that take a chemotherapy called Taxol, which many of you may have heard of, it is a chemotherapy that has a nasty side effect called neuropathy, which is nerve damage. That is cumulative, and it's obviously very disabling because it is something that can become permanent. So when you compare all women that get this drug, it's pretty clear that women that are Black get a lot more neuropathy than women that are not Black. And it's not because they got more or less or whatnot, it's because genetically they're more predisposed to it. Or conversely, like, if you look at people from Asian countries, if they take a drug called capecitabine, they can take a lot higher doses than women that are not Asian can take capecitabine. Again, that has to do with genetics. So the genetics of the person, the genetics of the tumor, all that makes such a big influence of what happens to the cancer story, the response to treatment and whatnot, and all this overarching genetic profiling of not just the tumor, but of the patient as well, will probably answer a lot of the questions of these health disparities between different ethnic groups, where, again, it's not one size fits all. I mean, and unfortunately, like, not just here in the US, but across the world, there are some ethnic groups that really just have a poor prognosis to begin with and are more prone to develop more aggressive cancer. So again, Black women, for instance, they get more triple negative cancer, and in general, they have higher mortality, even if they get the same access to care and treatment that women that are not Black. So there's really more than just socioeconomical disparities or access to care. There's this whole genetic profiling that needs to happen from both tumor and patient point of view that will hopefully one day explain why this health differences happens, and most importantly, help us customize treatment where we're optimizing cure and minimizing side effects.
Thank you, Ingrid. And I just again want to say that Komen is invested across health disparities in breast cancer, both through dedicated research in the health disparity space, training of the next generation to increase diversity from whether it's bench-based research all the way to physicians and clinical care, as well as, and importantly, community, state, and national level policy, support and guidance in making as it relates to really reducing significant health disparities that exist in breast cancer.
And Jennifer, and if I can make like a little shameful plea here to everybody in the call. I mean, at Vanderbilt, we have a lot of people obviously very interested in this health disparity. So we have a lot of cohorts of patients that we're following across not just the state, but across the whole southern area. So I would encourage patients or family from patients with breast cancer to really seek us out to see, can I donate blood? Can I be part of these registries? Because without these tissues, without this knowledge, without this long-term follow-up, it's going to be very hard to tease out what these differences are. So we have a number of different cohorts going, both through the epidemiology folks like Dr. Wei Zhang and Dr. Zhao Shu, who are actually, Dr. Zhao Shu, I think is on the call, like she's hidden there somewhere, but also like looking at patients that have BRCA mutations or other mutations that may be minorities as well. And Dr. Pietenpol is actually heading a lot of those efforts. So again, if you really have nothing better to do, please seek us out to help us like be part of this registry, donate blood, donate stuff, because that's really the only way we're going to find out these differences and do better.
Thank you, Ingrid. So in the last few minutes for the panel, and I want to be certain, Dawn, I think to leave 10 minutes for the question and answers in the chat for everyone who's just been patient through this panel. I just want to ask each of the panelists, what excites you the most? What does the future hold? You are all experts and highly engaged every day, not only in research, advancing new drugs, but taking care of women and men with breast cancer. And what are you most excited about? And maybe Skip, I'll start with you.
So important topic. I think we've always talked, and I've always told every breast cancer patient I've had the good fortune to meet to say no two women are alike or men and women are alike, and that every case is different. And now, as our panelists have confirmed through the DNA testing, through the liquid biopsies, through what we're able to analyze around the biology of a person's cancer, we in fact know that every case is different. So I'm most excited about the individualized approach. I'm excited about whether, women need to be, we treat a lot of women with hormonal therapy for 5 or 10 years because we're not sure which one of the 10 or 20 need it. What's the best first therapy for a patient? I mean, all these factors are really in the biology of the tumor, and we now have the tools to really understand that biology better than ever.
Great, thank you. Erika.
So I was, Skip, of course, stole mine, which he often does. So I give him a me too, but then I'll say the new drugs. From the antibody drug conjugates of getting smart enough to, put a chemo molecule on the back of an antibody and make sure that chemo gets exactly where it needs to go. Or another recent example is narrowing a tyrosine kinase inhibitor, a drug recently approved to Tucatinib, where we used to have a lot of rash and diarrhea with drugs in that class, getting smart enough to kind of engineer that molecule where it only hits its target and doesn't cause as many side effects. I think that some of these new drug advancements are going to be very important for patients, not only for outcome, but for tolerability.
Ingrid.
So me too, number 3, like a lot of the new drugs I think are exciting. I think the antibody conjugates really have an incredible future just because we're already seeing home runs, not just in HER2 positive disease, but in triple negative. And I think more and more we'll see more of these drugs come about. So again, less toxicity, better efficacy. Having them translate to the earlier setting may afford us the opportunity of have less people develop recurrence and consequently, cure more people. I think immunotherapy has a long way to go. I think I'm in principle very excited about that. But again, cautiously optimistic because I think we haven't narrowed it down yet. The best partner, the best patient, whatnot. But obviously that is very exciting because when you see a durable response with that, it's really remarkable. So I think both immunotherapy and antibody conjugates and some of these very neat targeted drugs to some driver mutations like HER2 are obviously very exciting stuff. And I think I'll stop at that.
Ben, you
said
to
final
come up
word.
with something completely original now.
Well, yeah, because I agree with all what was said, and it's, obvious areas of mine. But I would add the unique thing to me, and one of the things that got me to Vanderbilt that was just incredible, is the expertise in how we're going to disseminate and implement all of these great findings. So it's one thing that we at academic centers can do this to treat patients with the latest technologies, diagnostics, and therapies, it's another thing to push this out for the whole world to really also benefit from. And I think we all recognize that this goes into somewhat of disparities, but even amongst academicians, a lot of people are not necessarily, thinking about these. And so when you look at new drugs and how long it takes for targeted therapies to become widely disseminated and implemented, it can take up to a decade. That's got to change. And I think now with the internet and new technologies, it's going to change. And so another super exciting area for me in the world of breast cancer therapy and diagnostics is the ability to really make the world a much smaller place so that we can then also affect change for everyone, not just a few people.
Thank you all. I mean, really tremendous. And like I said, we're all so fortunate here in this region to have these unbelievable experts who have international reputations in the discovery, treatment, and care of breast cancer patients. So thanks to all four of you for really contributing this evening. Dawn, how do we want to do the question and answer? I see a number of them in the chat.
Yeah, I think Madeline was going to read those, and I just kind of want to echo too. I know that we didn't have time to go over each of our panelists' bios, but that link is in the chat, and so if anybody is interested, I just want you to know you can go to one of our web pages and actually get everybody's bio. But I'm going to ask Madeline to start with the questions.
All right, Ms. Dawn, can you hear me?
We can.
All right, so our very first question, do we have any new tools to monitor reoccurrence? And on that same note, can a liquid biopsy be used?
Is that directed toward anyone? I'm happy to take that.
So I don't know how, Jennifer, I guess,
Yeah.
if you want to say who it goes to. So none of them are specifically directed towards anyone, so I'm just going to throw them out there.
Ben, why don't you take that?
Yeah, I think that is a great question, and is in fact the hope of this new technology. But have we actually definitively proven that? Probably not. We're getting close. There are a number of studies looking at this, including one that we're doing at Vanderbilt with colleagues across the country. But one of my heroes in this business, Dan Hayes, who's the godfather of what we call biomarker development, often says that a bad test can be just as dangerous as a bad drug. And I really try to live and adhere by that philosophy. So we really have to prove that these tests are as good as we think they are. Otherwise, we could end up doing a lot more harm than good. And that's kind of where we are right now. Just like a drug trial, we're doing a biomarker trial.
Perfect. So next, a little different, we're going to talk about the move that we are kind of transitioning to for oral chemotherapy. So do you guys see this as an obstacle because insurance is going to pay different rates, depending on what type of chemotherapy they are needing to use?
Skip, did you want to?
Yeah, this is
yeah. You can put on your president of ASCO hat.
Yeah.
In part.
And I was going to say this in answer to the health equity question too. It's a struggle. This, the reimbursements methods and oral versus IV and all the various influencers in that business, pharmacy benefit managers and the like, really a struggle. ASCO and Komen and a number of national organizations have really been pushing at the congressional level and the state legislator level for oral parity bills. It shouldn't make a difference in terms of how the drug is administered. And we've got to push this forward and make this something that's a fair playground across all the country. It's already a legislative law in a number of states, and we just need to make sure that's true everywhere. But one thing is the legislators at the state and the national level do not like to carve out a disease. They do not like to pick cancer over arthritis, over cardiovascular, over diabetes. But this is a setting where these are, a different urgency in terms of these medicines that are being given. Not that all diseases are important, but nothing is more of a life-threatening event than hearing that you've been diagnosed with cancer. And we've got to continue to push that message with our legislators.
Perfect, thank you. So Ms. Ann asked, with so many of our resources today currently being directed towards COVID-19, are any of you worried about breast cancer research funding, either on the short-term level or on a long-term level?
Jennifer, what do you think there? What's your sense from
Yeah.
being the cancer center director? What's your sense on the NIH situation and where funding will roll?
I would say in terms of funding for cancer research and breast cancer research, the federal budget remains the same as pre-COVID, but where we've really seen the impact, tremendous impact, is in the funding we receive from nonprofits and the funding we have received from other sources, which have been terribly impacted economically by really everyone sheltering in place. And so that's where we've seen major impact. And it's, I think, why, especially from the standpoint of Susan G. Komen, we can't thank everyone enough for their support and to ask everyone to keep supporting the cause, because these are challenging times for foundations. I don't know yet what our prediction will be in terms of, depending on what happens to the economy, whether we see federal budgets get impacted in the next several years. And that would have a really negative impact on breast cancer research. As you can imagine, breast cancer is such a high incidence cancer, and we actually have a fair amount of the national portfolio is focused on breast cancer. So that could have an impact depending on what happens in the economy. I don't know if Ben, Ingrid, or Erika or Skip, you want to add to that from your purchase, what you're seeing.
I will just make a comment that we need to push on this, because through some of the ASCO talks with congressmen, they echo the same sentiments you just did, Jennifer, but also health and wellness has never been so front of mind for America. And so we've got to take advantage of this pandemic to push the initiative that if you don't have your health, you don't have anything. And that's the one place where we've got to take advantage of this crisis.
I may pipe in with one additional thing. Oh, sorry.
Go ahead, Erika.
I may pipe in that, trying to always see the silver lining of everything, that one thing that I've been really encouraged through COVID-19 is an attempt of kind of decentralizing some parts of clinical trials, sponsors becoming a little bit more lenient on local lab checks, or doing, some things not having to come to the trial center. And we know that for clinical trials that can be a burden on patients, having to get to a place where they can get that clinical trial. So I'm hopeful that that's something that we can take out of the backside of COVID and make clinical trials more accessible for more people.
And we've already seen a lot of that with telehealth possibilities. I mean, it's amazing how much farther now we can reach for second opinions. I mean, even if people don't come for a clinical trial, at least they listen and we can help them find one closer to where they live, whatnot. So I think that the telehealth was something that was always in the horizon, but it was very slow paced and not that much of a priority. And all of a sudden that went boom. And I think I echo what Erika just said. I think there is some silver lining where hopefully some of what we're experiencing now will be retained and will continue to have us reach farther to patients that cannot travel 5-6 hours to see us, but can still see us via telehealth and whatnot. So I think all that may be a positive spin on that.
And the other silver lining, as Erika was mentioning, of COVID-19 is actually an awareness truly of the power of discovery and the importance of science to deal with the human health issues, whether, people probably before this, Skip, I was thinking when you were talking about antibodies, a lot of people are probably thinking, it's the cocktail of antibodies to neutralize COVID. I mean, these are now things that are part of everyday discussion, vaccines and neutralizing antibodies, which weren't before. The one concern I think that many of us, though, in research and academics have, there are many young investigators who have really suffered during this time of COVID because of shutdowns in laboratories across the country and inability to do their research, and it actually impacts their next steps of their career. So we are really focused, and I know Komen's made a lot of attempts to say if funding does get limited, they're still going to try to invest in the next generation, early career investigators, keep this pipeline of people interested in developing drugs and finding new discoveries. So that's one thing that we are concerned about.
And actually, speaking of disparities, like, I don't know if you saw this, but there's actually research coming out now showing that women's research have gone downhill because of the need to stay home and take care of kids that couldn't go to school. So there's a disparity now between men publishing and women publishing because of that need. So it is what it is.
Do we have time for one or two more questions? Because there are several great questions in the chat.
I think we're going to have time for at least one more. So someone had mentioned earlier, I know we talked at the very beginning about different types of breast cancer. So someone had a question about inflammatory specifically. So kind of going back to the basics of breast cancer, I know a lot of us have heard that a lump is a symptom or a sign of breast cancer. A lot of people don't realize that there are actually many other warning signs. So I don't know if someone wants to talk very briefly about, you know, the different types of warning signs and kind of, you know, knowing your normal and where to go from there.
I could probably take that one. So yes, there are many things that look like breast cancer, and there are several different warning signs. By far, the most common thing that happens to somebody that gets diagnosed with cancer is honestly they have no symptoms. It's picked up on a mammogram. And that's good because typically those are smaller tumors, whatnot. But aside from that, the obvious things are a lump in the breast, a lump in the armpit, a distortion of the breast looks bigger or smaller and looks different than it did before. Any discharge, either bloody or non-bloody, coming from the nipple, any dimpling of the breast, any change in color of the breast. Something that it's a common misconception to go back to the whole inflammatory story is the concept of mastitis, right? Like a woman presents with like symptoms of mastitis. The breast is hot, is red, it hurts, it's you know, it's swollen and whatnot, and it looks like mastitis. And physicians usually would give antibiotics for that or whatnot. But the point is, if you're not pregnant or breastfeeding, it is not mastitis, it's breast cancer. And that's an important concept because a lot of primary care physicians and whatnot are not aware of that. And they'll keep, like, you know, forgive my French, dicking around with it. And then it becomes too late because this is a disease that is very unique, is very different than your regular breast cancer, regardless of subtype, where one day you don't have it, the next day you do. And it can evolve very quickly. It can metastasize very quickly. And it's not the same response as other cancers. It's like a different beast, and it's very, very, very aggressive, and it has to be treated very quickly. So it has to be recognized very quickly. So one key point is, if your breast looks inflamed, like it's infected, it is breast cancer unless you're breastfeeding. And that's a really an important distinction. If you're not breastfeeding, it's not mastitis. It is breast cancer, and that needs to be checked out. So, and yes, inflammatory, there's a big urgency to that because it is an incredibly aggressive form of breast cancer that unfortunately still kills a lot of people today.
Gosh, what a great hour that we have had. I know personally, I've been sitting over here taking notes. I have learned a lot tonight, and I just want to thank each of our speakers and panelists for their expertise and for taking time to share that with us tonight. I don't know that we got through all the questions, so what I'm going to tell you is that we will go through those, and we will make sure that there's an answer to all of the questions, and we'll get those back out to everyone. And last but not least, I just again want to thank our partners in this tonight. We could not have done this without you. So to LifePoint, to Nashville Yards, to Pfizer Oncology, Seattle Genetics, and Immunomedics, we thank you. We appreciate you being here and making this possible, and to each of you for taking time tonight to be here. If you have any questions, feel free to reach out to us. We are at Komen Central Tennessee.org. It's long, spell it all out, but that's our website. And again, I just want to say thank you to everybody for being here. Have a great night.
Thank you.
Thank you.